Hi students. I'm making this video, recording my lecture basically for our cell cycle and mitosis notes. So this is the first slide on the PowerPoint. Basically the cell cycle is the life of a cell and mitosis is how cells divide. So the phases of the cell cycle, it consists of an interphase, which if you see here is everything from G1 to DNA synthesis or S phase and G2. And then the mitotic phase or the cell division phase is just this little part here. We'll go into more detail on those in just a second. The functions of cell division, so basically reasons why cells divide at any point in time. Sometimes it's for reproduction, so single-celled organisms can use mitosis to make two new organisms, basically. Amoebas, for example, do this. You take one amoeba, cell divides, and then you have two amoebas. Growth and development, so for multicellular organisms, growing and developing is because of cell division. All cells, all humans at least, and lots of other organisms start out as a single cell, divide in half, two cells, those divide in half, four cells, etc., until we become the trillions of cells that we are right now. And then tissue renewal and repair. So your skin cells, for example, die and have to be replaced frequently. When that happens, a skin cell dies, it needs to be replaced. So a nearby healthy skin cell divides by mitosis to replace that dead skin cell. Not every single tissue in our body can be replaced, but many of them can. But brain cells, for example, just can't be replaced. But skin, bone, muscle, blood, all that stuff can be replaced. And it happens, new cells are made by mitosis. All right, so cell division is the process of mitosis and cytokinesis together. Overall, both of those things together make the whole beginning to end of cell division. Cell division is an important part of the cell cycle. It results into genetically identical daughter cells. Now, this is important because if you have a skin cell that needs to be replaced, you want your skin cells to have the exact same genetic material as the one that died, basically. There's another type of cell division that doesn't make identical daughter cells, and we'll talk about that in a different unit. But for now, mitosis makes genetically identical daughter cells, meaning they should have the exact same DNA as the parent cell and each other. So cells duplicate or replicate their genetic material. This is DNA replication, which hopefully you've already taken notes over. Before they divide, your DNA in those cells replicates to make two identical copies. This is so that both of our new cells will have an exact copy of the DNA. Chromosomes are basically how our DNA is organized in our cells. So all eukaryotic cells store genetic information in chromosomes, which is basically just our very, very long strands of DNA wrapping up around some proteins and condensing into smaller, more organized ways. They get wrapped up into chromosomes. And the picture below is showing you chromosomes. Each of those little blobs kind of looks like a chromosome. If you unwind it, is very long strands of DNA. Different species have different numbers of chromosomes. That's just based on their genes, how they organize their DNA into different chromosomes. So humans' DNA is organized into 46 total chromosomes. You have 23 pairs of chromosomes. You get half of a pair from your mom, and you get half of the other pair from your dad. So you have 23 chromosomes from each parent, and each parent gives you the same type of chromosome, and we'll get to that in just a second. So chromosomes are composed of a complex of DNA molecules and then wrapped around proteins. The proteins are called histones. That's not a huge important thing you need to know, but just FYI. Chromatin is what the loose DNA in proteins is called before it condenses into a chromosome. So chromatin condenses into chromosomes, so they organize themselves before cell division happens. DNA exists as a single long double-stranded molecule, and each individual molecule gets condensed into a chromosome. Each unduplicated chromosome contains one DNA molecule, which may be several inches long. It's like trillions of nucleotides long, though. And so the fact that we can actually see it, it's a very large molecule. And in every single one of your cells, there's inches worth of DNA, basically. A karyotype is basically just this picture down at the bottom of someone's chromosomes. Scientists can take chromosomes out of cells and organize them. And when they do that, it makes a karyotype. So a human karyotype would show the 23 pairs of chromosomes like it is below. You see there's pairs numbered 1, 2, 3, 4, 5, all the way through to 22. And then the third pair of chromosomes is the sex chromosomes X and Y, or X and X, depending on if you're a male or a female. Different organisms would have different numbers of, or different amounts of chromosomes. For example, dogs have 78 pairs of chromosomes. I'm sorry, they have 78 chromosomes total, and like that's 39 chromosomes, pairs of chromosomes, I think. Okay, so this is just a picture of how chromosomes are organized. If you see, there's the pair number 1. This shows you one of these chromosomes came from your dad, the other one came from your mom. And in this pair, one of these came from your dad, one of them came from your mom. So that's how you get 23 each. If you count them individually, you have 1, 2, 3, 4, 5, 6, etc., 46 total, or 23 pairs. Okay, so a diploid cell, this is a new vocab term. A diploid cell has 2 sets of chromosomes. Di stands for 2, so 2 full sets of chromosomes. Every cell in your body, except for sperm or eggs, is a diploid cell. So brain cells, skin cells, blood cells, they're all diploid cells and have all of your chromosomes. So a human has 46 chromosomes total. So every single one of your diploid cells has 46 chromosomes. In a cell in which DNA synthesis has happened, you're going to have copies of those chromosomes. So here, this is one chromosome, and it's sticking to its identical copy. This is right after DNA replication has happened. So same here. The reason they look like 2 or little X's is because this half is one chromosome, and this is the exact copy of that chromosome, and they're held together in the middle by a centromere for a little while until they get pulled apart in cell division. So here's another picture. So chromosome duplication, which is basically the exact same thing as DNA replication, because remember, this is a long strand of DNA that's just condensed to make a chromosome. This is like the one that you got from mom. This is the one you got from dad. It's one of the 23 pairs. After DNA replication, they look like this briefly, right before cell division, because I need one exact copy of this chromosome in each of my 2 new cells that's about to be made, and I need one exact copy of this chromosome in each of my cells that's about to be made. This is how our cells make sure that they get all of our DNA. So these are homologous chromosomes, the pairs where the chromosomes kind of look like the same shape and size, and they actually carry the same genetic information on them. They are called homologous chromosomes. So for example, if this was pair 1 in a human, this would be the one you got from mom. Maybe this is the one you got from dad. They carry the same gene. Maybe this is the section of this chromosome that carries the gene for eye color, and your mom has given you the gene for brown eyes. Then over here in this exact same place and section is the gene for eye color. This just happens to be on your dad's chromosome that he provided. And maybe he has the gene for blue eyes. You have both of them in your genetics, in your genetic code, but perhaps you have blue eyes because it's dominant. And we'll talk about dominant recessive another day, but that's kind of how that works. Before cell division happens, we need a copy of both chromosomes because technically they're all part of your genetic code. So you need a copy of mom's chromosome and a copy of dad's chromosome, for example. When you have copies of identical chromosomes stuck together at a centromere, they're called sister chromatids until they are pulled apart. But then you see this whole X, these are still homologous chromosomes. The purple and green are still homologous to one another because they carry the same information. And homologous, like homo means same, homologous just means it carries the same information. Okay, so here's the overall cell cycle. In every cell's life, it basically goes through these steps. Whether it's a cell that never divides and replicates like a brain cell, they're basically always stuck in this G1 phase. But cells that do divide, like skin cells, the cell starts in G1 and it grows and becomes larger. Then it does S phase where it replicates its DNA. Then it grows some more and makes copies of all the organelles because just like the two new cells need copies of DNA, they need copies of every other part of a cell that helps it function. And then you start cell division. And within cell division, there are 4 individual phases, which we'll talk about in more detail. And then the last step is cytokinesis, where the cells actually divide, the cytoplasm divides. And again, we'll talk about that in more detail. But then you got 2 new cells and they both start the cycle again, especially if they're skin cells, because those are always replicating frequently. All right, so the first phase of the cell cycle is interphase. And interphase makes up about 90% of a cell's life. And this is the growth or G1, S, and G2 phases. All 3 of those things together make up interphase. This is when the cell is growing. It's going to copy its DNA in the S phase, and then it's going to grow some more and get ready for cell division. It's also just doing its regular job. It's just being a skin cell, if that's the case. Or if it's a bone cell, it's just doing bone cell's job. Or if it's a muscle cell, it's just being a muscle cell doing the job that muscle cells do while also growing, replicating DNA, and then getting ready for division. So some key features of a cell in interphase is that its DNA is loose. It is not chromosomes yet. It's chromatin, loose, uncondensed DNA. It has a nucleus with a nuclear membrane intact. There may be some centrosomes. These are an organelle specific for cell division, and they only appear during G2 right before cell division is going to happen. So those are the key features of a cell. It's how you can look at a cell and know that it's an interphase. It has a nucleus, it has uncondensed DNA or chromatin, and it has these centrosomes appearing. Then we start with mitosis, and mitosis is 4 individual phases, which we'll talk about in just a second. Somatic, this is another new vocab word. Somatic cells are any cell in your body that is not a sperm cell if you're a male or an egg cell if you're a female. So we're talking about skin cells, blood cells, muscle cells, lung cells, liver cells, brain cells. They're all somatic cells or body cells, but the technical vocab term is somatic. These are diploid cells. They have all 46 of your chromosomes in them. And many of these types of cells, somatic cells, divide by mitosis. Now, like I said, brain cells don't, but skin cells do, for example. Blood cells do. Each new cell is going to receive 1 copy of every single chromosome. That is why DNA replication happens. And then after mitosis is over and cell division is over, it produces 2 new cells that are genetically identical, because hopefully these chromosomes were perfectly copied. So you see here we have this organism only has 6 chromosomes, for example, 3 pairs. DNA replication happens and you have the sister chromatids attached to one another by a centromere, that circle in the middle. Then after mitosis, you have 2 identical cells. They should have the exact same chromosomes as each other, but also as the parent cell over here does. So they should be identical to one another and the parent cell. The first step of mitosis is prophase. In prophase, the first thing that happens is you see here our chromatin is now condensed into chromosomes. So it doesn't look like it did in interphase before. We have individual chromosomes. There, you see, if you can kind of tell, there's two attached to a centromere. So each of these is a chromatid, and each of the bigger noodles is a chromosome. This one has 1, 2, 3, 4 chromosomes total. You will start to see spindle fibers come out of those centrosomes. These spindle fibers come into play later, but they start to form in this phase of mitosis. It's hard to tell right now, but the nuclear membrane is starting to disappear. The nucleus actually completely disappears before cell division, or in this phase of cell division, so that eventually our DNA can spread to do two different sides of a cell to be divided. The second step or phase of mitosis is metaphase. So now, as you can see, our nuclear membrane is gone. There is no nucleus right now. These chromosomes, as you can see here, have been lined up into the middle of the cell by the spindle fibers. So the spindle fibers have moved our chromosomes to line up in the middle of the cell. Now, one reason or one way to remember that is that meta, the prefix meta, means middle. So if the chromosomes are lined up in the middle of the cell, then it is during metaphase of mitosis. And these spindle fibers attach to the centromeres and actually push them there. These spindle fibers are made out of proteins, and they kind of push and pull the chromosomes until they line up in the middle. And as you can see here, these are the identical copies of chromosomes still attached to one another at the centromere for now. The third phase is anaphase. So now our sister chromatids, or the identical copies of every chromosome, are getting pulled apart from one another. Each of these was an identical copy created in DNA replication. If this was a human cell and we were able to fit it in the picture, you'd have 46 chromosomes on this side and 46 chromosomes on this side. But in the picture, it's pretty much impossible to show that. So here, our organism only has 4 chromosomes total, or 2 pairs. But by the end of anaphase, these chromosomes migrate to the sides of the poles of our cell. And then this is one whole copy of chromosomes, and this should be one whole copy of an organism's chromosomes. The last phase of mitosis is telophase. And now you can see our chromosomes have been pulled to each side. They're starting to loosen up again. They're not as condensed as they were before. Because our nucleus, so you can see our nuclear membrane is starting to form again around both of our new separated copies of DNA. All right, our spindle fibers are disappearing because they've finished their job. These centrosomes are going to go away. And so we have 2 new nuclei forming in our 2 new cells. Now, at the exact same time as telophase is creating 2 new nuclei and our chromosomes are starting to loosen up and decondense back into chromatin, cytokinesis is happening simultaneously. So at the exact same time as telophase, the last step of cell division, which is not technically part of mitosis, is happening, where the cell is pinching off and the cytoplasm is actually dividing. Here's what mitosis would look like in a plant cell. It's not too different, except for the fact that they have cell walls, and so that makes cytokinesis a little bit difficult. Yeah, okay, I want to make sure. We'll go. Oops. All right, there we go. Okay, so you have prophase. Prophase. You can see the chromosomes are not condensed completely yet, like they are over here. You still have a nuclear membrane, is pretty visible. Here, this is like in between prophase and metaphase. Our chromosomes are condensed. Our nuclear membrane is starting to loosen up and disappear. Now you can see the spindle fibers pushing these chromosomes into the middle of a cell. Here in anaphase, you can see the 2 sets of chromosomes being separated to opposite poles. And then here you can see the nucleus is starting to form. These are starting to condense back or decondense back into chromatin or loose DNA. And in the middle here is forming a new cell plate. So in cytokinesis for animals and plant cells, it's a little bit different. So cytokinesis is the division of the actual cytoplasm. Mitosis is just the division of the nucleus and all the DNA. And then the last step of cell division is cytokinesis, where everything in the cell, the cytoplasm, the cell membrane, everything divides in half. So as you can see here in animal cells, basically this pinching happens. It creates what's called a cleavage furrow, but there's some proteins, they're called actin filaments. They basically start to pinch off the cell from one side all the way through to the other, and eventually they separate. You can kind of see that over here too. In plant cells, because of the sturdy cell wall, they can't just pinch off and become two new cells. So basically what happens is in between the two new nuclei that are created, a new cell wall gets formed right in the middle of them to make them two separate cells. And this is called a cell plate that gets put in between the two cells. So here's a little bit different. So basically you have any cell that doesn't have a cell wall is going to do it like this, where they just pinch off and the cell membranes and the cell cytoplasm break off until they become two separate cells. But with a plant cell, the cell wall doesn't pinch. It's very sturdy. It can't be just pinched off. So a cell plate is built in between the two new cells. So you can see we definitely have smaller cells here, and over time they have to grow until they're about as large as the original cell was, but they just grow next to each other essentially. Here are some pictures of what plant cells look like under mitosis. Eventually you would need to be able to look at these and decide like what phase is this cell in. It's in prophase. The chromosomes are starting to condense a little bit. This is definitely prophase here as well. We've got condensed chromosomes in our nucleus. This one, all the chromosomes are lined up in the middle of the cell, so that would be metaphase. Then here we have our chromosomes pushing away from each other, going to opposite sides of the cell, so that would be anaphase. And then here, this is a good one, we have telophase starting to happen. Our chromosomes are decondensing, there's nuclei starting to form, and then we've got telophase and cytokinesis starting to happen right here in between these two smaller nuclei. So hopefully you can see the different phases based on what the chromosomes are doing in these plant cells. That's the end of the notes. If you have any questions, you can email me or come to Zoom tutorials. Make sure you took notes. If you need to go back, rewind, replay anything, please do that. Have a great day.